Alright, so far we have studied the three types of protein fibers that are found in the connective tissue. Next up is the ground substance. What we studied in the previous chapter about ground substance is that it is a gel-like amorphous liquid that is found in the extracellular matrix. But now we're going to study the details of what that gel-like amorphous liquid is made up of, and why it is so gel-like, watery, and fluid. Or in scientific words, you could say it is hydrated. Why is that? So the reason behind that is because it has very water-loving or water-attracting components. So these would be your GAGs and proteoglycans, especially your glycosaminoglycans or GAGs. So the primary component of the ground substance that is water-loving and that keeps it hydrated would be glycosaminoglycans. These are also called mucopolysaccharides. Polysaccharides would mean a chain of sugars, and then muco would be mucus-like, so hydrated, and it's basically a chain of sugars. It's a polysaccharide, and that's what GAGs are. And as we all know, that where sugar is present, that area becomes concentrated and it tends to pull in water towards it. And same is the case for GAGs. So any place, any area, any connective tissue where GAGs are in abundance, they would tend to pull in water towards them. Why? Because sugars are very water attracting. They make the tissue more concentrated. So GAGs are the reason behind the hydrated form or gel-like or amorphous form of the ground substance. Is because they are polysaccharides and they tend to attract or pull water towards where they are present or where they are being secreted. And as we learned earlier, that they are secreted by the fibroblast in the connective tissue. Now, GAGs are also polysaccharides that contain an amino group. I'll show you that in a bit, but let me just go over some examples. So some examples of GAGs would be dermatan sulfate, heparan sulfate, keratin sulfate, chondroitin, or hyaluronic acid. I've mentioned this term before. Chondroitin is a GAG that is abundantly present in cartilage and in your articular joints. So any person who is suffering for, let's say, osteoarthritis, you would give them chondroitin and glucosamine supplements to slow down the process of the disintegration of the cartilage. So anyways, you don't need to remember all of that. You just need to know what GAGs are and some examples of them. You don't need to remember the areas. We will cover specific areas as we go along the series and in their own chapters. We will specifically mention which GAG is present in which area if need be. But one important glycosaminoglycan is hyaluronic acid. This is also called hyaluronin. Hyaluronic acid, of course, like other GAGs, attracts and binds to water, and it does that by having all these sugar chains that tend to attract the water towards them. So we study that GAGs also have an amino group, and inside for the hyaluronic acid, we have this amino group over here, and because of the fact that it is an acid, it also has a carboxyl group right here. But what's important are the polysaccharide chain, or its quality of being a mucopolysaccharide, which tends to attract water. So water will be moved in towards wherever hyaluronic acid is present, and it would make that particular tissue more hydrated and more viscous and more have more mucus, and that would help. The hydration and the increased water content would then help that particular connective tissue for the functions of lubrication and transport, and also cushioning as well. So lubrication, transport, and cushioning. So lubrication, for example, between two articular cartilages, you might see in synovial joints, and in transport, of course, in the interstitial fluid that we'll discuss shortly. All right. So proteoglycans are just GAGs that are surrounded by core proteins. Okay, these are just GAGs that are surrounded by core proteins. So for example, proteoglycan. Proteoglycan we mentioned in the first chapter, and when we were studying the basement membrane, is that proteoglycan is present in the basement membrane, and what it does is that it determines the porosity of the basement membrane. Another example of proteoglycan would be Agrican. Agrican, it is present in cartilage, and what it does is that it binds to hyaluronin or hyaluronic acid. And as it binds to hyaluronic acid, the hyaluronic acid tends to attract water towards it in the cartilage, which results in lubrication, for example, if it's present in the cartilage of an articular joint. Another component of the ground substance would be multi-adhesive glycoproteins, adhesive again, junctioning sort of glycoproteins that allow binding of two structures together. So for example, laminin that we've studied before, it is a binding site for integrins, and another important multi-adhesive glycoprotein would be fibronectin. This is a binding site for collagen. So fibronectin would be a protein present in the connective tissue that binds to a cell, let's say using integrin of the cell, and then on the other side it would bind it to collagen and allow the cells to bind to the collagen. All right, so we've made this whole fuss about ground substance being hydrated, and it is saccharide rich, so it pulls in a lot of water. But the question arises as to where that water is coming from. And the answer to that is where all the water in all the tissues in our body come from, and that is the blood, through blood vessels. And same is, of course, the case for connective tissue. So here is an image. So in this picture, we see a blood vessel that starts off as an arteriole, and then it narrows its lumen and becomes a capillary right here, and then it goes along and becomes a venule, and is connected to this connective tissue right here, which of course contains the cells and then the ground substance, the extracellular matrix, the ground substance, and the protein fibers. The ground substance is the hydrated gel-like amorphous liquid that contains the hydrated part, that contains the watery stuff. This watery fluid is called the interstitial fluid right here, and this interstitial fluid is basically water, and it dissolves ions. Where do they come from? They come from the arteriole. As it narrows down its lumen, so this lumen becomes narrow inside the capillary, then there's pressure built up right here. This pressure is called the hydrostatic pressure, all right? And this hydrostatic pressure allows the water to move out of the capillary lumen into the extracellular matrix inside the connective tissue. And as it moves out, it only takes along with it the dissolved ions that are present inside the water. It does not, the endothelium, of course, does not allow any proteins or any cells to pass out of the capillary lumen, so only the water and dissolved ion comes out, and they come inside this extracellular matrix space, and then it is called the interstitial fluid. Now this fluid could be used for many purposes, like for example in cartilage, inside other tissues that need lubrication or transport. Now let me just give you a quick example of its function. So for example, this right here is the capillary epithelium, and then this is the connective tissue, right? And then this connective tissue might just be connected to some other tissue, like for example the muscle, right? As we know, muscles are very demanding. They have this constant need for other ions and nutrients, and then they're constantly giving out the carbon dioxide that needs to be transported out. And this exchange of materials requires a medium, and that is provided by the connective tissue extracellular matrix that contains the interstitial fluid. So the interstitial fluid, fresh interstitial fluid, is always coming and after the exchange, it is going or moving out into the back into the venule, or it could actually also go into the lymphatics. And the way it does that is through colloid osmotic pressure, just osmotic pressure. So in the case of colloid osmotic pressure, the water gradient of, let's assume, the lumen, the water gradient of the lumen in the venule would be lower, so water would move out of the interstitial fluid into the lumen of the venule, and then is transported away from that connective tissue. All right, so let's discuss some clinical correlates. So GAGs, we discussed that a component of the extracellular matrix is the ground substance, and ground substance is made up of three major things: the GAGs, the proteoglycans, and the multi-adhesive glycoproteins. And GAGs we discussed in detail. Now GAGs, of course, are also degraded in our body, and the way that happens is through enzymes, and the defects of these enzymes, like for example mutations, can lead to these specific pathologies: Hunter, Hurler, Sanfilippo, and Mercure syndromes. This topic is these pathologies are mostly concerned with biochemistry, so you might study them in biochemistry and medical genetics. Bacteria that can hydrolyze hyaluronan disrupts mucus barrier and possesses greater invasiveness. Of course it does. Like for example, you have a tissue right here, right? And you have this mucus barrier right here, and it contains little molecules of hyaluronan that are pulling all the water in. And then there's a bacteria that can digest this particular mucus barrier. Then of course it's going to have a greater invasive power. So that's that. And then edema that we discussed when we were studying mast cells. Remember, we studied the cells of the connective tissue, and one of the important cells of the connective tissue were the mast cells. And we studied that what mast cells do is that they bring about the immediate hypersensitivity reactions or allergic reactions. And the way they do that is by releasing secretions such as histamines or serine proteases, and chemotactic factors, etc. So secretions that cause the inflammation, such as histamines, these make vascular endothelium more permeable to leak interstitial fluid. The more permeable the vascular endothelia, the more water would be moved out, and it would cause a buildup of water outside or in the extracellular matrix, which would lead to edema. All right.